Next, we move on to ActiveTransport. In ActiveTransport, the way you're going to identify it is that you're going to see a solute moving against its gradient. Sometimes we'll refer to this as up its gradient. So it's going to be moving from an area where everything's really loosely spread out, where there's only a little bit of a solute, to an area where it's highly concentrated, where there's more of a solute. This is energetically unfavorable. So this isn't a process that happens naturally. It's going to require the input of some form of energy. We're going to see this energy put in two different ways. The first, we're going to see it directly put in our cellular form of energy, which is a molecule called ATP. In our second type of active transport, we're going to see that sometimes we use another gradient, so one molecule going from an area of high concentration to an area of low concentration, as we saw in passive transport, to sort of pull along a second molecule against its gradient. So from an area of low concentration to an area of high concentration. So these are going to be our two types. So our first type, we're going to directly use energy. And in our second type, we're first going to have to set up a gradient of something else that we can then release to pull our molecule through. So here we can see both forms of active transport. So in this example on the left, I want you to note that this pinkish colored protein is our protein at timestamp 1 and then timestamp 2. This isn't two separate proteins. Same deal for the purple ones here on the right. So what we see happening here, so we can see that when we start out, we have a low concentration of these hydrogen ions, so that's the H+, or these white triangles on this side of the cell in our intracellular environment. And we have a high concentration out here in the extracellular environment on the outside side of our cell. So using this first integral protein, you can see what happens is that we let the hydrogen go into our transporter or our carrier. We use ATP to create a shape change and push it through. So it's going up to its gradient, and we're making this area more concentrated, this extracellular part. So that is an example of primary active transport because we're directly using ATP to go against the concentration gradient. Now in the second example, we can see that we have that same gradient for hydrogen, so low here in the cytosol, high in the extracellular environment. But we can see that we have another molecule labeled as well. So sucrose, one of our carbohydrates, a sugar. We have a high concentration inside the cell in the cytosol, and a low concentration outside in the extracellular environment. But what do we see happening through our transport protein? So we see co-transport happening, right? We have hydrogen and sucrose both going into this transporter, moving the same direction. Both hydrogen and sucrose are being moved into the cell. Now, this is going to be active transport because if we look at that sucrose, it's going from an area of low concentration to an area of high concentration. Now, we don't see a direct input of energy here, but what is actually happening is that the hydrogen wants to flow down its gradient, so it wants to flow from high to low, and we let it do that, so it's going from high to low, it's moving with its concentration gradient, and as it does that, it takes a sucrose with it. So this protein is a co-transporter, allowing for secondary active transport. We're going to go through an example of each of these that is important in cells. So our first, probably honestly, our most important transporter is something called the sodium-potassium pump. Now it's an antiporter, and it pumps two ions, sodium and potassium, in opposite directions. It does this actively, so we sometimes will also see this referred to as a sodium-potassium ATPase to tell you that it's using ATP. But anytime you see the word pump, we only use the word pump when we're using ATP. So that's also a good signal that it's an active transport situation. So it moves 3 sodium out of a cell and 2 potassium into the cell every time it does its job. And it does this by breaking down ATP, using it for energy. So this is an example of primary active transport because we're directly using ATP. And actually both these molecules would be going against their gradients. The gradients aren't drawn on in this example, but this is just showing us the full picture of how this would work. And this is more complicated than you should expect to see in this class. And then I will expect you to know. But this shows us kind of the time progression of how this protein would work. So the way it works is that it has basically these slots for sodium on one side, 3 sodium bind in, then ATP comes in, we break a bond in ATP, and that pushes those 3 sodium to the outside of the cell. Now it has slots facing the outside of the cell, and they wait until 2 potassium molecules bind in. When these 2 potassium molecules bind in, the protein changes shape and shoves those into the cell. But whenever we see this in this class, we'll probably just see a little cartoon with 2 arrows going opposite directions for sodium and potassium. But you should be able to recognize if you see sodium and potassium going opposite directions across a membrane, that that's likely happening due to a sodium-potassium pump. Our example of secondary active transport, we're going to talk about the sodium glucose symporter. And a symporter is just another word for a co-transporter. So like our example with hydrogen and sucrose, what happens is we start with a concentration gradient of sodium. So this is our pre-existing gradient. We have a high concentration of sodium in our extracellular fluid here, so outside the cell. And here we see we have a low concentration of sodium inside the cell in the cytosol. And so sodium moves down its concentration gradient. And if that were the only thing happening, we would expect this to be a type of passive transport. However, we're also looking at these glucose molecules. Now glucose, there's very little glucose in this example outside the cell in the extracellular fluid, only maybe this one right here. But glucose is still moving into the cell. So we can see that by the little faint arrow here on the glucose. So we can see that glucose is moving from an area of low to high. And that's basically because as these 2 sodium molecules bind in, they take a glucose molecule into the cell with them. So that is an example of secondary active transport because we're using the sodium gradient to move the glucose. Now, bulk transport is going to be our last type of active transport. We'll see 2 types, exocytosis and endocytosis. And bulk transport is used when we need to move really big molecules or many molecules or particles that are basically too big to just go through a protein. So this is a type of active transport, but we're not going to see how the energy is applied here because we're basically zoomed out a bit too much to see that. This is usually going to be transporting things like proteins and polysaccharides. Like I said, big stuff. So the basics that you need to know are basically just the direction of bulk transport every time we see one of these words. So in exocytosis, we're moving material from inside the cell to outside the cell. And in endocytosis, we see something moving from outside the cell into the cell. Now, in both examples, we can see something pretty cool happening with our membranes here. So because the membrane is fluid and neighboring molecules aren't totally attached to each other, we can basically create little bubbles of membrane. And when they hit our larger cell membrane, they break open and push everything to the outside. So that's what we see happening in exocytosis. We can see a little bubble of membrane inside our cell, which we'll see is called a vesicle. So this is a vesicle. So this vesicle has a lot of something inside it, probably some protein that we want to get outside of the cell. We can see it comes up to the membrane, fuses with it, and as that bubble breaks and the walls of the bubble become part of the cell membrane, that protein that was inside the bag is released outside the cell. We can see the opposite thing happening in endocytosis. So we actually start with our plasma membrane, some proteins or whatever, this solute that we're going to pull in through endocytosis, they start to collect on the cell membrane, and it starts to dip in. So it invaginates. Invaginates just means that it starts to sort of cave in, form a little divot here. So it forms a bubble like this, which then breaks off, and we can then carry whatever it is that we've just taken in somewhere else into the cell in this little bag. Okay, and so that is bulk transport. So this slide is just a summary slide of the different types of transport you're responsible for knowing. So far we've gone through passive transport. We had 3 types. Simple diffusion, which is directly across the cell membrane. Osmosis, which is where we have water move instead of our solute because the solute can't get across the membrane. Then we have facilitated diffusion, which is where we have our molecules going through usually a channel protein, sometimes a uniporter. Okay, and all of those are examples of passive transport. They don't require energy. We're going to see molecules moving from areas of high concentration to areas of low concentration. The most typical example we see is this facilitated diffusion example, where we're going through usually a channel protein. Now we've gone through active transport, where we see that we're using energy either directly in the form of ATP, where we can break a bond to release energy, or we have a second type of active transport where we use the gradient from some other molecule to carry our molecule through. Or we have bulk transport where we use energy in some kind of nebulous way to allow our membrane to fold in endocytosis, or little vesicles to merge with our membrane and push our molecule out in exocytosis.